Hi, good morning. My name is Dr. Jason Wasfi. I'm an associate professor at Harvard Medical School. I'm a clinical cardiologist. I direct our program in cardiology outcomes research at the Mass General Hospital Cardiology Division. I am also the chair of New England CEPAC. I am going to introduce our voting panel today. We are really looking forward to an incredibly important session, which raises a lot of really interesting and important issues. I will sort of start by saying that I have no updates to my declarations of conflict of interest. I'm going to go through the voting panel in series, and I'd ask you to introduce yourself and briefly state your affiliations and sort of tell us if you have any updates to your COI disclosures for today's session. I'm going to start with Dr. Austin Fracht.
Good morning. I'm Austin Fracht. I'm a health economist with appointments at the Boston VA and the Harvard School of Public Health.
Megan Golden.
Hi, I'm Megan Golden. I'm the co-founder and CEO of Mission Cure, a nonprofit patient advocacy group focused on pancreatitis, and I have no conflicts to disclose.
Thank you, Megan. Rebecca Kirsch.
Good morning. I'm Rebecca Kirsch with the National Patient Advocate Foundation based in Washington, D.C. No conflicts to disclose.
Thank you. Next is our vice chair, Dr. Steve Kogut.
Morning, everybody. I'm Steve Kogut. I'm a professor and also vice chair of the Department of Pharmacy Practice and Clinical Research at the University of Rhode Island College of Pharmacy, and I have no updates to my conflicts of interest to note.
Thank you, Steve. Don Creese.
Good morning, everybody. I'm Don Creese. I am New Hampshire's consumer advocate, which means I represent the interests of residential utility customers in my state. But I'm really here as a patient and family advocate, having raised a 23-year-old awesome woman to adulthood, despite the fact, or because of the fact that she lives with cystic fibrosis, a chronic illness, and I have no updates to my conflict of interest disclosure.
Thanks, Don. Dr. Julie Coopers.
Good morning, everyone. I'm Julie Coopers. I am the Vice President of Clinical at Alera Group, and I have no updates to my conflict of interest.
Thank you. Dr. Tara Lavelle.
Hi, everyone. I'm Tara Lavelle. I'm a health economist at Tufts Medical Center and on the faculty there, and I have no updates to my conflict of interest.
Thank you. Dr. Aaron Mitchell.
Hello, I'm a medical oncologist and health policy researcher at Memorial Sloan Kettering Cancer Center in New York City. No updates to my disclosures. I continue to have none.
Great. Dr. Brian O'Sullivan.
Good morning. I'm a pediatric pulmonologist at Dartmouth Hitchcock Health and professor of pediatrics at the Geisel School of Medicine at Dartmouth, and no updates to my conflict of interest.
Thank you. Joe Porter.
Good morning. My name is Joe Porter. I'm the Chief Strategy Officer at the New Hampshire Center for Justice and Equity, where I focus my work on health equity, and I have no additional or no disclosures to update today.
Thank you. Dr. Rishi Wadera.
Good morning, everyone. I'm an associate professor at Harvard Medical School and the Harvard School of Public Health, and also a cardiologist at Beth Israel Deaconess Medical Center. I have no updates to my disclosures.
Thank you, Rishi. So just for the whole group, it sounds like there are no updates to disclosures, but every member of this panel has previously made disclosures and have no conflicts of interest to disclose, which are defined as individual healthcare stock ownership, including people's member in the household, in any company with a product under study, including the comparators at the meeting in excess of $10,000 during the previous year, or any healthcare consultancy income from the manufacturer of the product being evaluated. So at this point, I'd like to call on, we have an amazing group of experts to enliven the discussion today, and these include both patient and clinical experts. So I'd like to start with the patient experts, starting with Dr. Todd Durham.
Good morning, everyone. I'm Todd Durham, the Senior Vice President of Clinical and Outcomes Research at the Foundation Fighting Blindness, and you see here my up-to-date disclosures of conflict of interest.
And thank you for joining us. Julie Grutzmacher.
Good morning, everyone. I'm Julie Grutzmacher. I'm from Prevent Blindness. I'm the Director of Patient Advocacy and Population Health. I run a training program for the visually impaired and blind in advocacy skills and storytelling. And I've had a good number of RP patients, about 20 of 250 trained.
Thank you. And then any conflicts of interest to disclose?
No conflicts of interest.
Thank you.
If you don't mind, I'm going to quickly jump in. Hi, everybody. I'm Sarah Emond. I'm ICER's president and CEO. And I wanted to just quickly mention that, as you can see, we take potential influences on judgment and conflicts of interest very seriously. Given this is a small condition with a small number of companies working in this space, we tried really hard to balance different perspectives in our clinical experts. And we are so grateful for Dr. Russell and Dr. Mahajan being with us today. And I'm just inviting them to say hello and give a little extra context to how they relate to some of the manufacturers in this space, both under study today and some potential competitors, just so our audience has a broad view of their potential influences on judgment. Dr. Russell, might I invite you to go first, please?
Sure. My name is Steve Russell. I am a full-time clinician scientist at the University of Iowa, and I have been involved in investigating a number of drugs to treat patients with retinitis pigmentosa. Most prominently, I was a PI in the Luxturna trial, which was the first approved drug in this space. I have no additional conflicts of interest to discuss, but would be happy to comment on anything that anybody has any concerns about.
That's perfect. Thank you, Dr. Russell. And Dr. Mahajan.
Hi there. Good morning. I'm a professor of ophthalmology at Stanford University. I'm a vitreoretinal surgeon and scientist. My PhD is in biochemistry and signal transduction. I'm involved in a number of human gene therapy trials for genetic and non-genetic eye diseases. As such, I advise, I'm a paid advisor to a number of companies, including Nanoscope Therapeutics, which is the company whose product we are discussing today.
Wonderful. Thank you both. All right, next slide, please. I am pleased to be joined by several members of ICER's team and esteemed collaborators today. You'll be hearing from Dr. David Rind, our Chief Medical Officer, who served as the senior management team lead for this review. Dr. Anil Makam, who served as an evidence author, who's one of our collaborators from UCSF. And Dr. Marina Richardson, who is the lead health economics modeler for the review today that we'll be hearing from a little bit later when we talk about comparative value. Next slide, please. We always like to start our conversation by remembering why we're here. We're here today because there are patients with retinitis pigmentosa who would like to have an effective treatment for their condition. And we heard this story from someone living with RP. Life is this is a life of constant adjustments and adaptations. Because once you think you have one level of sight down, you have to learn how to accommodate all over again. The eyes are very important. You pick up all those gestures in the room or just visual cues. You know you lose that when you have an eye condition, an impairment. It changes the quality of your life. There's not a lot of jobs you can do. People don't want to work with you. They don't understand you. It alters your life course journey too. So wherever you were succeeding, you have to now change course and decide, okay, how do I adapt? Next slide. When we're faced with a new treatment in the pipeline, potentially getting approved for a condition, we have to think about what happens on the day it receives FDA approval. There might be celebration. for the advancement that it represents for treating a condition that affects patients. But we have questions that we ask. What are the risks and the benefits? How does this new treatment fit into the armamentarium that we already have? What are the reasonable prices and costs to patients, the health system, and to the government? And what lessons are being learned to guide our actions in the future? Next slide. That's what we're doing today. We're doing that today because we can have a conversation about how to affordably introduce a new intervention to the healthcare system while managing that introduction in a way that doesn't continue to impact families who are struggling to afford health insurance. We know that we have had stories, and I don't think you can go a week these days without hearing about the impact of rising healthcare premiums on employers and on families and on taxpayers. We know the wards of Chicago went $80,000 in debt because their twins were born premature. We know that Sherry Foy was forced into bankruptcy because she had to have surgery to remove her colon. And the Bucks of Arizona tried to see a doctor and were turned away because they hadn't paid their bills. And so they ended up seeking emergency care. And most recently, the National Bureau of Economic Research actually quantified what happens when health insurance premiums go up. It leads to an increase in unemployment, and it leads into an increase in spending for families. Next slide. What we do here as part of this work with our external appraisal committees like New England CEPAC is we foster a conversation about how we can be focused on the patient in the room with their doctor while still remaining focused on all of the other people in our healthcare system who need affordable access to care. That is the underpinning of the mission of ICER, is that we can have that conversation and we can be sure that we are paying fairly for new therapies in a way that doesn't harm the unseen people in our healthcare system. Next slide. So, ICER, we are the convening group, and we are a nonprofit organization doing health policy research. We have 3 external appraisal committees made up of experts. You heard from the New England CEPAC and their amazing expertise this morning. And we also have a Midwest and a California group. Next slide. We like to be transparent about our funding. We just asked everyone else to be, so we are too. You can see a list of every organization that's given us money in the last 12 months on our website. All of our research is funded by nonprofit philanthropies, which include Arnold Ventures, the California Healthcare Foundation, the Patrick and Catherine Weldon Donahue Medical Research Foundation, and the Peterson Center on Healthcare. Any money that we do take from manufacturers, health plans, PBMs, or provider groups supports a policy summit that we hold each year to try to tackle one big issue in the drug pricing space. Just this week, we put out our report on how to have affordable access to GLP-1 medicines for obesity, if you want to check that out. Next slide. So how did we get to this point? We started several months ago. About 7 months ago, we gathered together to decide on our research scope, but that meant that we needed to talk to patients and patient advocacy groups, clinical experts, the manufacturer, and other critical stakeholders. They informed that scope. It was available for public comment. We went through a process of analyzing and synthesizing the evidence, and that was led by Dr. Makeham from UCSF, and then the model, the economic model, was developed by Dr. Richardson. Our report went through expert review before it went out for public comment, and we're deeply grateful for those listed here who provided their perspective on the draft. After we received public comment, we revised our report and published it as an evidence report. That evidence report forms the basis of the conversation today with our New England CEPAC, which will go through a deliberation of the evidence and a vote for what they think the evidence represents in terms of net health benefit and other benefits and contextual considerations. Next slide. So I've mentioned this idea of thinking about value. So what does that mean? Snapshot of our value assessment framework. In green are the things that are represented in a systematic literature review, an evidence synthesis, and an economic model, where we're looking to see what the evidence tells us about how much better patients feel and maybe how much longer they live if we were looking at a life-limiting condition. And how does it get them back to their activities of daily living, their own goals, as well as the costs that the new intervention represents to the system. But that also includes potential cost savings that might be represented by a new therapy. But we'll spend a lot of time today hearing from patients and clinical experts about the stuff in blue. Benefits beyond health and special ethical priorities are incredibly important considerations when we think about the overall value of a new medicine. But guess what? Sometimes they're really hard to quantify. So hearing from the patient groups directly and their stories that they hear from patients, hearing from clinical experts about what they see when they're trying to treat patients with retinitis pigmentosa, that's going to be an important consideration for us in our overall judgment of value today. Next slide. All right, you've made it through the opening remarks. Congratulations. Next, we will have the presentation of the clinical evidence from Dr. Maycomb, followed by the presentation of the economic model from Dr. Richardson. We'll hear from the public, and we'll have a discussion as part of those public comments. After a lunch break, the New England CEPAC will deliberate on the evidence that they've seen, and they'll vote on the comparative clinical effectiveness, and they'll talk about and comment on those other benefits beyond health and special ethical priorities. After a break, I'll be back to moderate a conversation with all of the experts that have been with us today from the patient and clinical community, as well as the manufacturer and some representatives from the health plan space, so that we can have a conversation about how we translate this evidence into policy that will lead to meaningful, affordable access for patients. We'll close with some reflections from the New England CEPAC, and we expect to wrap up today around 4 o'clock Eastern. With that, it is my pleasure to introduce Dr. Maycomb to walk us through the clinical evidence.
Great. Good morning, everyone. Thank you, Sarah, for the introduction. I'm going to be presenting the clinical evidence. Next slide. Oh, sorry. Let me go back. All right. I would like to recognize the important contributions of Avery, Belen, and Sol for their outstanding contributions to the report. We're part of the clinical research team and are all employees of ICER. My institution, on my behalf, received funding for my contributions on this report, and we have no other conflicts of interest to disclose. Retinitis pigmentosa, or RP, is a group of inherited diseases that causes progressive degeneration or loss of photoreceptor cells. It affects about 1 in 4,000 individuals, with a prevalence estimated in the US of about 80 to 110,000 people. It's not considered a lethal disease by itself, and the age of onset and rate of progression vary and in large part depend on the underlying genetic mutation. Regardless of the underlying cause, the hallmark symptoms of RP are loss of night and peripheral vision. However, RP affects many different symptoms of vision, including difficulty with transitions in light, as well as seeing colors. And for the 12% or so of people who have advanced RP, they have symptoms related to central vision loss, which can range from being able to count fingers to no light perception whatsoever. There are no cures or disease-modifying therapies for RP. And for individuals living with advanced RP, usual care typically consists of vision aids, vision rehabilitation, and managing various eye complications such as cataracts. Optogenetic therapy is a novel approach to treating RP. And this consists of inserting or injecting a gene therapy into the eye that inserts light-sensitive proteins known as opsins to allow non-photoreceptor cells in the retina to begin acting as photoreceptors to potentially restore vision. And because it aims to work at reprogramming the visual pathways, it's considered mutation agnostic and in theory should work across the full range of RP mutations. However, optogenic therapy is reserved for the treatment of patients with advanced RP with severe vision loss so as not to interfere with intact visual systems. Sonperidogene istaparvovec is the first optogenetic therapy that we anticipate will be considered for approval by the FDA. It's administered by a one-time intravitreal injection of the eye, which inserts the opsin protein MCO10 into the middle layer of the retina in the back of the eye, as shown here on the right, which largely consists of bipolar cells. We heard several key insights from our discussions with patients throughout the course of our report. People living with RP described a slow progression until more advanced RP when the quote-unquote series of losses became more rapid. People described many visual symptoms that vary day to day and affect regular activities, such as driving and reading. As such, people described psychosocial and emotional distress, especially about concern about progression to complete and total blindness. However, several people we spoke to discussed that with adaptation, that they still lead meaningful, productive lives. But we note that this was often contingent on socioeconomic status. People we spoke with were eager for treatments that restored greater visual functioning, such as restoring the ability to recognize faces and read, but also know that if they were completely blind, regaining some light perception was thought to be helpful. Before I jump into the clinical evidence, I want to spend some time discussing the outcomes of the pivotal trial. The primary outcome of the trial is best corrected visual acuity, or BCVA. And this is measured using a validated computerized tool that was specifically adapted for individuals with low vision, known as the FRACT. It's scored using the logMAR, which is the logarithm of the minimum angle of resolution, which is a mathematical transformation of the smallest visual angle that I can see, where higher scores, which indicate wider angles, reflect worse visual acuity. The minimal clinically important difference is a change on the logMAR of greater than 0.3, which translates to roughly 3 lines on the chart, which is shown here as a screenshot from the computerized tool. The logMAR ranges typically from 0 for 20/20 vision to a floor value of 2.25. This table shows the rough mapping of logMAR scores to key visual stages, which will be more important. My colleague Marina talks about the economic model. And these include better than counting fingers, and hand movement. Importantly, the stages of light perception and no light perception are not measurable because individuals at these stages are off the chart and cannot complete the test. Because RP has many visual symptoms, we'll be discussing 2 key secondary outcomes of the trial. The first is mobility, as measured by the multi-luminance Y mobility test. This was adapted by the manufacturer for individuals with low vision, and they test participants at up to 6 light levels, ranging from an overcast day to a dark night sky. Success at each level was defined as correct identification of a lighted panel through a Y-shaped obstacle course, as shown here on the right, 3 times. The minimal clinically important difference, as suggested by the manufacturer, was passing this test at least 2 dimmer light levels. The other secondary outcome we'll be discussing today is shape discrimination, as measured by the multi-luminance shape discrimination test, as shown here on the right. This is a novel manufacturer-developed outcome where they test participants at up to 5 light levels, ranging from a dimly lit room to a dark night sky. And success at each light level was the correct identification of shapes 3 times. And again, the minimal clinically important difference, as suggested by the manufacturer, was an improvement of at least 2 dimmer light levels. Now I'm going to present the clinical evidence. The evidence base for sonperenogene is comprised of the RESTORE trial, which is a 27-participant phase 2b randomized, double-masked, multi-center sham-controlled trial of a 100-week duration, with change from baseline outcomes assessed at 52 weeks. The trial tested 2 different doses of sonperenogene, a low and a high dose, injected into a single eye versus a sham protocol. The trial included adults with advanced RP with a visual acuity of greater than 1.9 logMAR in the study eye. The average age of participants was 56. Nearly all were white. and the average logMAR score was 2.2, which roughly translates to being able to detect hand movement. This slide shows the main results for the primary outcome of visual acuity, or BCVA. And what this shows is that both the low dose and the high dose treatment groups showed a statistically significant improvement in the change from baseline of logMAR of over 0.3, which again is the minimal clinically important difference compared to the sham group, which virtually showed no change. In a secondary responder analysis, 7 or 39% of treated participants were considered responders on the logMAR score versus 1 of the sham control group. This slide shows changes in visual acuity over time. So on the vertical axis is change from baseline and the log score, where higher values on this axis indicate improvement in visual acuity. The 3 treatment groups are color-coded, so the sham group is in green, and the low and high dose treatment groups are in red and blue respectively. On the horizontal axis are the different time points when visual acuity is measured. And what this shows is the 2 treatment groups more clearly separate by week 24. By week 52, which was the results I showed in the previous slide, both treatment groups were above the minimal clinically important difference threshold, which is shown in this dashed gray line. By week 76, the high dose group peaks, and both treatment groups decline by week 100 under this threshold. And as you can see with the sham group, there's virtually no change throughout the study period. Because it's a very small trial and these averages can be skewed by just 1 or 2 patients, this figure or slide shows individual participant response for visual acuity for all 27 participants. So what's shown on the vertical axis are the masked identification codes for each participant. MCO, again, is the abbreviation for the opsin. HD and LD are high dose and low dose respectively. The horizontal axis shows the logMAR score, where the lower visual acuity and the floor is 2.25 shown on the left, and improved visual acuity is as you move to the right. And what this shows is that 10 sonperidogene treated participants had detectable improvement, but there was a wide range from 0.04 to a dramatic response of 1.83. And what this slide also shows is that 8 sonperidogene treated participants and 6 sham control participants had no detectable improvement. But importantly, all of these individuals were at the floor at the baseline, meaning that if they had a change, it might not have been able to be detected due to the nature of the test. And what the slide also shows is sham participant number 8 had a major protocol deviation related to an incorrect recording of their visual acuity. So what appears to be a dramatic response, it was incorrect. And to complement the results of visual acuity, the slide shows the key secondary outcomes for the combined sonperidogene treatment group compared to the sham control group for both mobility and shape discrimination. The outcome of interest is bolded, which is the change from baseline for both outcomes. And as you can see here, the combined sonperidogene group had a numerically greater improvement for both outcomes. And for the combined group, they had an improvement of 3 light levels for mobility compared to 2 for the sham control group. And for shape discrimination, the combined treatment group had an improvement of nearly 2 light levels compared to 0.2 for the sham control group. Neither were statistically significant, but this was a small trial. And in a responder analysis, nearly around 60% of the combined treatment group were considered responders, compared to about 20 to 30% of the sham group. But again, important to note is that some of these participants were near or at the ceiling for both scores at baseline, so they did not have the opportunity to be considered a responder. This slide shows the post hoc composite responder analysis at week 52 for all 3 visual outcomes. The combined sonperidogene group showed a response in at least 1 outcome for all 18 participants compared to 5 or 56% of the sham control group. Over half of the sonperidogene treated group showed a response in at least 2 visual outcomes compared to 1 of the sham group. And then just 1 treated participant showed a response in all 3 outcomes compared to none for the sham control group. And the definitions of the response are here at the bottom. In terms of safety, most treated participants experienced a mild to moderate adverse event of the eye compared to about 2/3 of the sham group. And these were largely detected on eye exam, which showed inflammatory cells in the eye, elevated eye pressure, and conjunctival hemorrhage, which is a tiny bleed of vessels in the conjunctiva at the front of the eye. There was a similarly low use of topical steroid therapy to treat these complications between groups at week 52, and there are no serious harms or death detected during the course of this study. There are several important controversies and uncertainties to contextualize and interpret the evidence. First, that this is an emerging evidence base for a new biotechnology for a rare disease, so it consists of 1 small trial of relatively short duration that's not yet made publicly available. There are many visual symptoms in advanced RP, and these outcomes are difficult to measure. When they are measured, they can be hard to interpret for the floor and ceiling effects that I shared before. And there was some internal inconsistency across all 3 visual outcomes that we noted for some participants. Additionally, long-term durability is unknown for this therapy. Some experts who work on in the optogenetic space question the biologic plausibility since they're surprised by the benefits seen compared to other related opsin therapy and were worried by the choice of animal models. We note the potential for unmasking of the sham control procedure, as well as possible reporting bias as some mismatch between secondary outcomes and outcome data that was shared with us. And then lastly, as the trial tested treatment of only 1 eye, the evidence for treating both eyes is uncertain, and we note that the potential gains may be less in treating both eyes than in treating a single eye. We received several public comments during the course of our report. The manufacturer thought we were being overly worried about the durability of treatment effect as well as safety, as these were established in mouse models. However, experts we spoke to all agreed that longer-term evidence in humans would be preferred, and in the absence of such data, preclinical evidence in larger mammals was preferred that more closely resemble the visual system in humans. We also heard that to emphasize health equity considerations for adaptation to severe vision loss, which includes individuals with more limited financial means, time flexibility, and social support. And we agree with this comment, and we note that that a potential benefit beyond the clinical evidence that I just shared is a potential for health equity gains if a treatment can restore vision for people who are least able to adapt to severe vision loss, with the potential for less caregiver burden. In summary, sonperidogene appears well tolerated and could potentially have clinically meaningful improvements in vision, at least in the short term. Our enthusiasm for the evidence was tempered by several uncertainties, including an emerging evidence base with a single small trial, unknown longer-term harms, challenges with outcome measurement, interpretation, and consistency across all 3 outcomes, and unknown durability of effect with uncertain biologic plausibility. As such, we rate the evidence for sonperidogene compared to usual care for adults with advanced RP as promising but inconclusive. And with that, I'm happy to take any questions that people may have.
Thank you so much for the presentation, and congratulations to you and the whole research team. I already see a question from Austin, from Dr. Frecht.
Yeah, I have 2 related questions that pertain to what was shown on slide 29. I don't know if you could put it back up because it'll be easier for me to discuss
Maddie,
it.
are you able to go back to the slide?
29, and if not, I can do it verbally. But okay, So we have one patient, number 13, with the largest response as indicated here. And, you have to look awfully closely, I admit, but it looks like at baseline, they were starting at a slightly higher logMAR value. So my first question is there any reason to believe that slight increase at baseline, or I'm sorry, decrease actually, slight lower baseline score could relate to the large decrease overall at week 52? And I'll just ask my second question now, which is to what extent are the overall average findings driven by that one patient with the large response?
Thanks so much, Austin. So for your first question, was the dramatic response related to the fact that they were slightly above the floor value? It's possible. Again, with a very small trial, it's hard to know whether heterogeneity response was driven by baseline visual acuity. Again, because of the floor, it's hard to know where people started based on this outcome. So certainly people who didn't have as dramatic response may have had changes, just nothing that could be detected based on this test. And I don't know if there's anything more that we can shed light on the single patient. And to your second question, whether this changed the averages, certainly. I think with 18 treated participants, with one person with a pretty dramatic change, it can change averages at different time points. We did not calculate those averages because those were estimated from kind of mixed effects models that we did not have available to us.
Thank you. Megan Golden.
Thank you. I'm not a physician or a scientist here, so I might have misunderstood, but I thought that there was a slide that showed that 56% of the people in the sham group had improvement on something. And if that is correct, why would that be for a progressive disease like this?
Yeah, great question, Megan. I think the 56%, if I recall, is the composite responder analysis where responded at least on one outcome. So that was those outcomes were visual acuity or passing the mobility or shape discrimination. It's hard to know. The natural history evidence in advanced RP is limited. We do note that we heard from patients there's a lot of variability day to day. Two of the outcomes are manufacturer developed or adapted, and so the minimal clinically important difference as well as the reliability of these tests are unknown. So whether there was improvement just because of noise versus true improvement, I don't know if we would be able to say with great certainty.
Thank you. Steve, I'm going to get to you in one sec, but can I just interject a little bit on that because it's relevant to this response? Can you comment a little bit more on the MCID issue? It sounds like it's defined by the manufacturer and there's not a lot of evidence it's just curious in your discussions with patients, for example, did that seem like a reasonable MCID or any more contextualization that we have I know lots of conditions don't have an MCID, so I don't mean to be complaining, but just your thoughts about whether that's a reasonable, minimally clinically important difference.
Yeah, thank you, Jason. I will say for visual acuity, that's more known in the field. For the two secondary outcomes of mobility and shape discrimination, since these were novel outcomes, there's not a literature base for the MCID. These were more suggested. I think on face validity, an improvement of at least success at 2 dimmer light levels do make sense, and people described, a desire for improvement in light perception. I don't know if we would, whether it's 1 or 3. I don't think there's a strong guidance there.
Yeah, no, that makes sense. Steve, Dr. Kogut, Steve.
Yeah, you preempted my question, but I could put maybe a finer point on it. Am I interpreting the results correctly in that for the secondary outcomes, the MCID was not achieved? And then could you comment a little bit about You know, this idea or this notion of assessing effectiveness based on average results when there's such a clear difference between, you know, folks who responded and then those that didn't.
Yeah, so I'll take your first question on the minimal clinically important difference. You know, there's numerically greater response in the sonpridogene group. You know, for mobility, it was kind of above that threshold of 2, and for the shape discrimination, it was very close to 2. And maybe I'll turn to our clinical experts of Dr. Mahajan and Dr. Russell. If you had greater context of those tests. I know there's this is new in the context of this trial, but whether either of you had a way to kind of help people, you know, wrap their arms around this.
Yeah, we'd love to hear the perspectives of the clinical experts on that question.
Dinu, do you want to go first?
No, go ahead. And just to understand, the question is about the meaning of those specific tests. Is that fair?
Yeah, so I
Yeah,
think
I think so.
I think that the question has to do with the high variability between the responders and the non-responders and why there isn't a more graded effect. And of course, there's also the noise problem that you have. So there are a number of aspects that affect not only a small study, but also when you get into the high noise range. So one of the things that we don't know about this patient population that could explain a lot of variability is their genotype. So we're assuming that the response is uniform, because this mechanism, if it is effective, should be relatively agnostic to gene type, but we don't know that for sure. Okay, so that's a premise. So we need to keep that in mind. The second issue is that these are extremely advanced patients in terms of the degeneration of the retina. And so the question then becomes, if you, let's say you have somebody who is degenerated, but they've retained enough enough of their inner retinal neurons, bipolars and ganglion cells, that they have enough viable cells that they can respond to gene therapy. In other words, they have to have a viable cell that can be transfected, as opposed to some of these patients who may be so advanced that they don't have enough viable cells so that they can be transfected. What you might see is what you see in this particular outcome plot, which is some patients respond very well and some patients respond not so well. So unless we had some idea of, for instance, how long the patient had been at no light perception compared to, you know, so if they were freshly at light perception or no light perception, as opposed to having been at that degenerative level for 10 years, say, then you might expect a significant difference in responsiveness, because you may have more viable retinal targets in the one population where you wouldn't in the other. So I just, you know, the fact that there is a non-responsive population, I think one of the things we just have to remember is that these are extremely advanced patients. So there may be a non-responsive group in there. And again, this is a phase 2b, so that makes sense. We're looking at dose ranging and, you know, kind of honing in on what patient population might be the best for a phase 3. Anyway, I'll stop right there. If you have any questions, I'm happy to answer them.
Yeah, can we pull up slide 29? Again, I think if we have that picture up, I can, the comments will be a little more clear.
Maddie, do you know if you can advance the slides or reverse the slides of this? Or Grace?
Yeah. So I agree with what Steve just said. So I would say, this is a patient population that nobody's ever done a clinical trial in because of their advanced vision loss. And so it's hard to benchmark this against prior experience of clinical trials because of the advanced vision loss. What I do think is really important to point out is a floor effect and what that means. We use a lot of jargon, so I'm going to try to move my hands around to explain the floor effect. So as someone had mentioned, there's one patient who is a little bit to the right and had a big improvement. And that suggests they entered the trial and their baseline was a little better than everybody else. And we see this big change. What a floor effect means is all of those patients without an arrow to the right, there's no way in this trial, or in many instances, people haven't figured out how to measure where they actually started on the left. So we can imagine that many of these patients, if we could draw a graph to the left, they might be showing a delta, but we just can't measure that in this kind of a test. So then we're stuck doing some of the other tests. And the manufacturer, as you guys mentioned, had made new tests. And so it's not possible to benchmark that against previous trials. But those new tests are asking in very, very low light levels, can patients make decisions about how to move around, which is sort of a distance thing. And then in a new test of objects placed in front of them, testing near vision, could you pick up a fork? Do you know that there's a glass in front of you? Do you know what's on your plate of food or something like that? Could they show improvement? I think the noise is quite reasonable. The patient variability of near tasks, mobility, reading letters, it's not unusual for patients to not consistently have the exact same behavior for all of those three tests. So it's very challenging, as people have mentioned. This is, it's a phase 2, like Steve mentioned. The patients are very heterogeneous. This is, in fact, gene agnostic because there's no categorization of the patients based on their gene. And as Steve mentioned, different patients, just based on their genetics, could be entering the trial at a different stage. They could be entering the trial with different levels of degeneration. And that might account for why we're having the floor effect. But the data should be noisy. And to me, that looks like a real trial. What is remarkable are the percentage of patients that did improve and how much they improved.
Can I just clarify, do you think that this phenomenon that you're talking about around the floor effect, is it correct to speculate, I guess, that if anything, this biases the trial to the null? If some of these individuals may be, I don't know what the right term is here, but maybe improving in ways that we can't detect from the logMAR score? Is it, I don't mean to put words in your mouth, but is it correct?
Yeah, so I think that's a correct sum that we're using a test that may not be measuring improvements because we don't have a good test when the vision is that low. And sometimes this happens, patients come into the clinic and they tell you, hey, I think I improved, or I'm able to do this, or I can't do that. And we don't have a test to measure it. And in those instances, you're listening to the patients and what they're telling you. With the sham, you have that comparison. There was a question earlier, why are sham patients improving? One can imagine somewhat of a placebo effect, maybe a training effect. And then we're just going to do our best in comparing the sham to treated and see where we land statistically.
Thank you. Megan, I think you have another question.
Yes, for Anil, on the limitation near the end that this is based on one small study with a short time frame. I'm wondering, to me, it seems like 100 weeks is actually quite long. As a patient advocate who's always trying to advocate for sponsors to conduct trials. They're telling me that it's not feasible to do one more that's more than several months. So I'm wondering if we're trying to hold the trials to a standard that's too high to be feasible.
Thank you, Megan. I think in the context of this treatment, it's a one-time lifetime treatment. So while 100 weeks, typically when we think about treatments, can be more reasonable, kind of thinking about what's the durability of a gene therapy over the course of a life. The average age was 56. So kind of projecting out, is it safe? Is it effective 10, 15 years out? Becomes more challenging. And we have, there's more experience now with Luxturna from, kind of more quote unquote real world or observational data past the trial. And I think this is a reasonable, this would be expected for a new technology for a rare disease, but just kind of noting that's just the nature of the evidence that we have.
Thank you. I think there's a question. We actually are doing great on time, so we have lots of time to have a really good discussion. Brian, Dr. O'Sullivan.
Yeah, just following up on that, it did look like there was loss of efficacy over time, if I read that graph right, that out by close to 100 weeks, there really was loss. And if we're talking about someone who's a one-time therapy to hopefully last 20 years or more, that may be significant.
Yeah, so what the figure you're talking about, the change in visual acuity over time, again, very small trial, so and a noisy outcome. I don't think we put a ton of stock in differences from each time point, although the general trends don't show a consistent improvement over the 100 weeks. Whether it was measured again and it was right at the threshold, I don't know, but there is a 3-year follow-up observational study for the individuals enrolled in this trial. So there will be data up to 5 years at some point, just it's not publicly available yet.
There's a comment or question from Dr. David Rind, the Chief Medical Officer Advisor. David, you want to go ahead?
Yeah, I was just going to say that I spent a lot of time staring at that graph, trying to decide whether I could predict into the future. And I really don't think it's fair to do that over those small number of patients and the up and down that it's doing over time. I think you could almost tell any story you wanted to about what the future would look like. It's also worth remembering that the graph actually ends past the 100-week at, I think, 126 weeks or 120 weeks. And there are very few patients in the graph at that point. So that data is it's very uncertain. So I would to Megan's question, I would say we always have this problem with a new gene therapy. We're talking about a therapy that's supposed to last years or decades, and we're worried about safety and we're worried about efficacy, and we're going to have to make decisions long before we find out what happens a decade from now. And that's just the nature of the beast. But it's still fair, I think, for us to point out that uncertainty exists, even if it's almost inherent in the therapy that there will be that uncertainty. It's good still to remind people that we're uncertain.
Dr. Mahan, I think you have something to respond.
Yeah, I'll just add just a few comments. I think the last round of comments are, you can't predict the future. We have the amazing Luxturna trial as our benchmark that we like to think about for gene therapy. But that was one very specific disease with a different vector, different patient population, et cetera, et cetera. So even though that's what we have, it's hard to benchmark against that because of the difference. There are some patients that were in a phase 1/2 trial that have shown sustained improvement at 5 years. Of course, that's not statistically significant. That's not the trial in front of us. But there is some optimism from that they maintain functional improvement. And then I will say with all of the human gene therapies, there is an issue of developing cataracts at a certain time point. And so at a year plus or minus, the patients develop cataracts. And I'm fairly certain this trial doesn't, none of the trials have removing or correcting the cataract as part of the trial, but discerning if that dip is actually from cataract development or loss of function is an open question. This gene therapy is unusual in that one of the protein components fluoresces. So you can actually take a picture of the human eye, and this is the only gene therapy where you can see that it's still making a protein. And so I don't think this is unlikely that the gene therapy is not being made as an explanation for that dip. And that's some of the patients where you can see that.
Any additional questions or comments? Maybe since we have a couple more minutes, Neil, let me just kind of, I know these evidence ratings are always really tricky, right? They're subjective and you have to sort of make a call one way or the other. In this case, the evidence team assigned a P/I rating. I mean, look, if you don't mind, let me just kind of push back a little bit. So obviously I can understand where the I is coming from, right? There's a lot of uncertainty about the MCID, about heterogeneous treatment effects, about how this would work over time, about how this would work in different genetic variants. But on the other hand, it sounds like this is like the first randomized trial done in this disease condition. I don't know how much it costs to run a trial like this, but I assume it's enormous. And I assume this is a small company. It probably has limited resources. This is a sham placebo-controlled randomized trial that shows efficacy. I mean, I think obviously as like an academic, I'd love a 1,000-person trial that followed people for 10 years and could analyze heterogeneous treatment effects, but that's just not feasible in this kind of situation. Why not give this a better rating than P/I? Maybe, I know this can be a struggle sometimes, so I'd love to hear more about the rationale for the I.
So
we had a I know, David, I don't know if you want to take this. We thought a lot about this.
I do to a degree. So we almost always, on a new therapy where safety is a concern, include the possibility of harm, which is what the P/I does. And it's hard not to do that on almost any new therapy with a new mechanism of action. For people who watched our discussion of a new medication for pain control at our last meeting, which is a much more standard randomized trial comparing drugs to each other, there was no particular evidence of new harms, but we know that on the order of 25% of new biologic therapies get a new black box warning after FDA approval over the next 5 years. So the rate at which harms come up that people didn't think they knew about is pretty high. And giving a rating, say, of C+ or C++ to this therapy would say we have high certainty that there's not harm, that and we talked a lot about this. Almost everybody agreed with this rating, but we had a lot of discussion about it. I'll say one thing about this, and it shows up in the report as well. This vector gets to the other eye, and the explanation to how it gets to the other eye is thought to be that it goes up through the optic nerve into the brain, across the optic chiasm, and back out to the other retina. Does that cause any problems inside the brain? No evidence that it does. Do we have high certainty, which is what a C+ or a C++ would say, that couldn't cause problems? I'm very hesitant for us to claim high certainty, despite how cool this therapy is.
and I'm sure a lot more to learn about the efficacy and safety of this therapy going forward. So thank you, and congratulations again to the research team, to David, to Anil, for the enormous amount of work that's gone into this report. We are exactly on time. I am very happy about that. Thank you all, despite a vigorous conversation. So I would like to introduce Dr. Marina Richardson. She is the Associate Director of Health Technology Assessment Methods and Health Economics for ICER, and she led the economic modeling for this project. Dr. Richardson. Marina.
Perfect. Okay. So thanks so much for the introduction, Jason. So I would also like to acknowledge the contributions of Ujang Lee and Marie Phillips, who supported the modeling efforts, and we have no conflicts of interest to disclose. So the objective of our analysis was to evaluate the lifetime cost-effectiveness of sonpretogene compared to usual care for the treatment of patients with advanced RP and severe vision loss. Now, to inform considerations of unmet need for RP in a quantitative way, we can use measures called shortfalls, both an absolute shortfall and a proportional shortfall. And shortfalls allow us to show the years of life adjusted for quality that are lost with RP compared to the general population without the condition. So you can see on both an absolute and proportional basis, RP has a substantial unmet need compared to other example conditions. Now, unique to this review was a patient pilot project where, in addition to ICER's standard process for engaging the patient community throughout the review, we conducted 3 focus sessions with 4 patients from the RP community to discuss the draft model analysis plan and draft evidence report. The aim of this pilot was to work with the patient community to ensure that their experiences and goals for treatment were reflected in the cost-effectiveness analysis. Now, full details of the impact of this pilot are included in the evidence report, but in short, the impact of these sessions included helping select the clinical outcome measured in and additional scenario analysis conducted. It helped with the data inputs that were selected and their validation, and really highlighted several important deliberative considerations. And I would also like to take this opportunity to thank the Foundation Fighting Blindness and Prevent Blindness for their help in connecting us with potential participants. Our methods followed standard methods as described in ICER's reference case for cost-effectiveness analysis. One item I'll note here is related to outcomes. So in addition to standard outcomes, we also included outcomes for years with vision better than counting fingers gained and years with light perception gained. And this was based on feedback from patients that in addition to improvements in vision, a meaningful outcome would be to prevent further decline in vision. Our model structure included 6 health states, 5 based on visual functioning and 1 deaf state. So the 5 levels of visual functioning represented are from the highest level of visual functioning as better than counting fingers and progressing to the worst level of visual functioning as no light perception. So in the model, patients started in one of the green health states from counting fingers to no light perception, and no patient started in better than counting fingers. Now, transitions between health states or staying within the same health state occurred annually, and patients could move to the deaf state from any state after year 1 of the model. Just to note as well, for simplicity, we haven't shown arrows that may jump more than 1 health state. However, this is possible in the model. Our model cohort characteristics follow the characteristics of patients from the RESTORE trial. And at baseline, patients had vision of counting fingers or worse. Again, a full list of our key model assumptions are included in the evidence report, but I'm going to highlight 2 important assumptions here. So first is related to treatment effectiveness. And And for treatment effectiveness, this was modeled based on a composite endpoint of best corrected visual acuity, mobility testing, and shape discrimination testing at week 52. So as you saw in the clinical presentation, the data were noisy. There were a small number of patients in the RESTORE trial. There were floor and ceiling effects, and in some cases, a lack of internal consistency between measures. So we did identify a priori that we would need a method to determine treatment effectiveness that considered all three key outcome measures from the trial. So in doing so, we classified patients as having improved, worsened, or staying in the same state. And we determined that if a patient improved or was at the ceiling of at least 2 of the 3 measures, they would be considered improved. Worsening followed the same rule, and the extent of improvement, if a patient did improve, that would be determined by the week 52 BCVA. The second assumption I'm going to highlight here is the treatment effect and the durability of that effect. So for this assumption, we assume that treatment durability would last for 5 years, followed by a progressive decline in visual functioning over another 5 years, at which point, being at year 10, treated patients would then return to the vision level of untreated patients. And this assumption was based on clinical expert opinion, suggesting that 5 to 7 years was a reasonable expectation of durability, given that there was only limited data beyond week 100 of the RESTORE trial. Just to note as well, we did run several scenario and sensitivity analyses to test alternative assumptions for treatment durability. Baseline health state classification of patients was the same for both treatment arms and was informed by confidential data from the manufacturer. And we calculated the baseline distribution using a weighted average of patients in the intervention and the control arm of the RESTORE trial. And then treatment effect at year 1 was then based on the composite outcome that I described a few slides ago, which classified patients as having improved, worsened, or stayed the same at each level of visual functioning at baseline. And then extent of improvement would then, as I mentioned, be determined by BCVA at week 52. And because that was a lot to illustrate how this composite measure works, I'm going to walk through a hypothetical example. So let's say that patient A starts in a health state of hand motion. And then at year 1, that patient experiences improvements in BCVA, reaching a logMAR of 1.75, improvements in mobility testing, and no change in shape discrimination. So because that patient improved in 2 out of the 3 outcome measures, and BCVA at week 52 represents a level of visual functioning of better than counting fingers, patient A would have been considered to have improved by 2 health states, moving from hand motion to better than counting fingers at year 1 of the model. Okay, and then what happens after year 1? So we have year 1 treatment effect informed by the method I just described. So patients in both arms then maintain their same health state until year 2, based on week 100 data from the RESTORE trial. And then after year 2, for the usual care arm, we assumed a progressive decline in visual functioning based on natural history data. And then at the top of the screen for sonperadagine, from years 2 to 5, patients maintain their year 1 health state. From year 5 to 10, patients experience a progressive decline in visual functioning, returning to the level of untreated patients at year 10. And then after year 10, progress at the same rate as usual care, according to natural history data. Moving into other key model inputs, we used an anticipated placeholder price for sonperadagine of $437,500, which assumes treatment is received in only one eye, the worst seeing eye. So this placeholder estimate represents half the midpoint of the range predicted by IPD Analytics because their estimate did assume treatment in both eyes. We also included a 6% markup given the therapy will be provider administered. Annual healthcare costs amounted to just over $19,000 annually, regardless of health state, and included inpatient, outpatient, and pharmacy costs. And also noting that these costs are inclusive of related and unrelated RP costs. And it was based on a retrospective claims analysis of patients with RP from the US. Direct non-healthcare costs, such as low vision services and devices, and indirect costs, such as impacts on productivity, were included in the societal perspective analysis. Health state utilities were primarily informed by a recent utility elicitation study conducted in the UK. So all health states have substantially reduced utility values, ranging from 0.54 in better than counting fingers, and decreasing progressively to 0.26 for no light perception. Okay, moving on to results. So treatment with sonpretogene resulted in improvements in quality of life and a greater number of years with vision better than counting fingers and with light perception. There is no anticipated impact of treatment on survival, and as such, there are no improvements seen in life years gained between patients receiving sonpretogene and usual care. For costs, based on a placeholder price, treatment with sonpretogene resulted in higher costs compared to usual care, which was due to the anticipated treatment costs and associated markup. There were no differences in non-intervention costs, given that there were no differences in direct medical costs with changes in visual functioning between sonpretogene and usual care. For incremental results, again, keeping in mind these are all based on a placeholder price for sonpretogene, the incremental cost-effectiveness ratio was 646,000 per QALY or EVLY gained. And the cost per QALY and EVLY gained were the same because there were no differences in survival. And as with all cost-effectiveness analyses, we performed a number of sensitivity and scenario analyses. The first one being a one-way sensitivity analysis, where we varied all parameters individually across reasonable ranges of uncertainty. The main drivers of the analysis were utility values for better than counting fingers and light perception, average age of patients receiving treatment, and the durability of treatment effect. Noting that the durability of treatment effect, we varied this parameter in the one-way sensitivity analysis from 2 years to 20 years. And all results remained above commonly cited cost-effectiveness thresholds, again, keeping in mind this is based on a placeholder price. For the probabilistic analysis, which is where we ran the model 1,000 times by randomly selecting a value for each parameter based on their probability distributions, we found that none of the 1,000 model runs reached cost-effectiveness thresholds of 50 to 150,000 per QALY or EVLY, again, based on a placeholder price. Key scenario analysis. So we did perform a number of scenario analyses in line with our value assessment framework modifications for single and short-term therapies. We ran a scenario analysis that under an optimistic scenario, assuming a 10-year durability of treatment effect. Under a conservative benefit scenario, we assume that there must be improvement in all 3 measures to be considered improved at 1 year. So that's a change from the composite measure that was used in the base case of 2 out of 3 measures improving to having all 3 measures be considered improved. And then finally, when we assumed a lifetime durability of treatment effect, cost effectiveness does improve, reaching $312,000 per QALY and EVLY, but again does remain above commonly used thresholds using a placeholder price. So to achieve cost effectiveness thresholds of $100,000 to $150,000 per quality or EVLY. Sonpretogene requires prices between $67,000 and $101,000 for treatment in one eye. There were several limitations to our analysis, primarily due to the clinical data available to inform treatment effectiveness. So the clinical trial data from the RESTORE trial was based on a small sample size, as mentioned, and there were limitations to the outcomes measured in the trial. There was very limited data beyond week 100 to inform estimates of treatment durability. However, I mentioned this was assessed in sensitivity and scenario analyses. We had no data to inform additional benefits that could be achieved from treating both eyes. And there was variability in health state utility values available in the literature, as well as validated by what we heard from patients during our focus sessions on the economic model. And again, this was assessed in sensitivity and scenario analysis. We received several comments in response to our draft evidence report. We heard concerns from the manufacturer about the baseline distribution of patients across health states not being the same between treatment arms, that the health state utility for better than counting fingers was underestimated, and that there was a mismatch between the placeholder price used in the clinical trial protocol of treating only one eye. And we made changes to the evidence report to address these top three concerns. And then for the fourth concern, the fourth bullet here on the slide, we heard concerns about assumptions for the durability of treatment effect. Given that there is a lack of long-term data on durability and having concerns with the evidence from studies in mice, we do believe that our 5-year durability of treatment effect with waning to year 10 was a reasonable estimate at this time. We did conduct, as I mentioned, several sensitivity and scenario analysis for alternative durability assumptions, including calculating threshold prices, assuming a lifetime durability of treatment effect. And with that analysis did find that the threshold prices would be approximately double those found in our base case if that lifetime durability was achieved. So in summary, patients treated with sonpretogene experienced gains in QALYs and EVLYs and a greater number of years with vision better than counting fingers compared to patients receiving usual care. At a placeholder price that assumes treatment in one eye, our analysis suggests that treatment with sonpretogene would exceed commonly used cost-effectiveness thresholds. When assuming that lifetime durability of treatment effect, cost-effectiveness would improve, but results remain above commonly used cost-effectiveness thresholds. With that, I thank you very much for your time and welcome any questions.
Thank you, Dr. Richardson, and congratulations on this whole body of work, and to you and your team. Marina, this was great. Let me just start by asking, and this is sort of a question for you in both of your roles, both as the lead for this, but also as your governance role for HCA methods at ICER. So we have to struggle now with utility weights that are for a condition that we've heard, and I know you've had a lot of discussions with patients and advocacy groups. People often adapt to their lives when they experience blindness. In many cases, these people had more vision and then lost it and could regain some of it. How should we consider the robustness of the utility weights that went into this model? Because I know that can be a struggle in health economics to consider utility weights for a condition where, in many cases, people are, have adapted their lives to living with blindness.
Yeah, it's a great question, Jason, and one that we talked a lot about as a team. I will just comment first on the evidence that we did use for the utility estimates. So this came from a UK study. So these were a set of vignettes that were put together and asked patients in the UK general population to respond to via what's called a time trade-off method. And these results were actually quite similar to results we saw in other studies from other countries across a number of different years. I think the oldest study we had for utilities was from 2001, and again, very, very consistent with that. Now, your comment about patients adapting to their condition over time, I think that's an important one to consider, and one that did come up even in all of our sessions with patients, that that they are very adaptable and accommodating to their situation. So they're very resilient individuals. I will say though, in the model, when it's used in the model, the important thing to think about is the changes that occur as patients move between health states, and how that differs between the intervention and usual care. And so because there's no extension of life in this case, I think I'm more comfortable with the use of the utility estimates, regardless of coming from patient population or general population, because of that fact, and because we're seeing differences in quality of life according to the treatment's effect on that across health states. And so that makes me more comfortable. I know. Yeah. David, did you have anything else to add on that front? Because I know it was quite a discussion.
These utility levels that we're seeing are extremely low. And I think many of them are coming from a population without blindness, seeing vignettes of what it would be to lose vision. And so as people adapt, you might get utility levels that are not so low. And the likely effect of that is that it would make a treatment for low vision look less valuable. The opposite, I think, of what people often are imagining when they think of what would happen when you talk to a general population. This happens in hemophilia, where people with hemophilia rate their quality of life much better than what people who are told about hemophilia would rate quality of life. And so a treatment for hemophilia based on utilities of people with hemophilia doesn't look as valuable as it does if you ask a general population. And so it's just important to remember that when you are asking a general population, you're actually getting a higher value, translating to a higher price for the therapy than if you ask people who had adapted to it. And that's a concern among patient groups, that because people have adapted, treatments can be undervalued. And so we don't see that here. We've got people describing very low utilities that as people adapt, they might not describe such bad utilities in those states. That said, as Marina's pointing out, she asked me because I was sort of stunned by how low those utility values were. A utility of 0.26 is the sort of thing that you'd see somebody who was lying in bed immobile after a stroke permanently. It's an incredibly low utility, and probably not what somebody who has adapted to blindness would describe.
Yeah, I mean, these are lower than advanced cancer and heart failure and stuff like that, as you point out. Thank you both for those great responses. We have a lot of hands up on the CEPAC, so I'm going to get going to Don Kreis.
Thank you, Jason. Dr. Richardson, as I said when I introduced myself, I'm here as a representative of the universe of patients and families that have an interest in inquiries like this. And I have to confess that in that capacity, I feel pretty acutely the fact that we don't have any actual patients here to meet and talk with today. I think this is the first meeting of this sort where that's been the case, and I can appreciate the difficulties that that resulted in that outcome. But that makes me really interested in the pilot project that you mentioned, which is talked about in one of the appendices. And so I'm wondering if you could maybe in a sentence or two tell me how that pilot project differs from what ICER would ordinarily do, and whether you think the pilot project was a success, whether that made any difference, and what impacts it might have had on the report itself.
Yeah, thanks so much for asking. So this patient pilot project was very exciting for us to do, and how the main way that it differs from our standard process is that it's very much focused on the cost effectiveness analysis and our model analysis plan. So in our typical patient engagement process, there's several opportunities for patients to comment through share your story forms, through small group discussions, commenting on each of our key deliverable. But this was, and so each of those opportunities really do still offer a chance for patients to comment and respond to questions that the team has related to the economic evaluation. But this was an opportunity for us to go directly to potential participants, so individual patients, talk about what we do. So what is health technology assessment? What is cost effectiveness analysis? What is a model analysis plan? And go through prior to posting our model analysis plan, saying, okay, this is what we're seeing, this is what we're hearing, this is what we're planning to do. Do you agree? Where are we going wrong? And so it really offers that two-way dialogue at an early stage to make sure their perspectives are included. And I mean, I'm quite biased. I think it was a great success. We did hear feedback. We asked for feedback from participants to see how they found the process, and it was quite positive. They found it very engaging. They found the opportunity to engage with other individuals with RP to be extremely helpful. And really, they felt like, a lot of the cost effectiveness analyses methods made a little bit more sense. In terms of impact on the model, I also think it was a success there. As I mentioned a few times on the slides, we in cost effectiveness analysis also report other clinical outcome measures besides QALYs and EVLYs. And so they helped inform us to say that maintenance of vision is important. So we're able to identify those additional outcome measures. We also heard about indirect effects. So when a patient moves from better than counting fingers to counting fingers, there's a larger impact on costs of low vision services and devices. So we are able to accommodate that in the model. And so those are just a few examples. I mean, you saw the report, but yeah, overall, I think it was a great success. I agree. It's unfortunate we don't have any patients with us right now, but I think Todd and Julie might be great people to go to on the call if there are specific questions there.
Super, thank you.
And we will hear more from them soon. We still have 3 questions. I'm trying to get to everyone. Awesome, Dr. Fract.
Yeah, hi. I'm going to try to triangulate across the 2 presentations on one question, which is we heard some speculation that the marginal benefit treating the second eye would be less than the marginal benefit of treating the first eye, and that was consistent with something David said about, I think if I understood correctly, some of the protein going through the brain to the second eye, right? We also heard, and so that was from the evidence review, from the cost effectiveness review. It was basically implied that the expected price of treating both eyes would be twice that of treating one eye. So if you put those 2 things together, the cost of per quality of a 2-eye treatment would be higher, much higher cost per quality than the 1-eye treatment. And I just wanted to poke at that, see if any of my assumptions are wrong or I misunderstood anything.
Okay, so I'm not sure I'm completely following, but I will. Okay, David has come off mute, so maybe he followed and would like to comment.
David, do you want to go ahead?
So I think our best guess, Austin, is that what you just said is right. The treating a second eye at the same price would make higher cost effectiveness numbers. You could imagine a story, though, given the small number of people treated. What if when you treat the second eye, what if every eye has about the same chance of getting a big jump? And if you treated the second eye, you were doubling your chance of getting so yes, I think that's right, but ultimately, I'm not sure it's right.
and we're still several
Are
multiple
we a
studies.
little more certain that the price would double, or do you think there's a chance the manufacturer would have a, a second eye half price type, I'm just, you know what I mean?
So what happened there, and Marina alluded to this, is the manufacturer, I think, fairly noted that we had initially doubled the price and were giving the increase in benefit of a study of one eye. And so we said, okay, we don't know what's going to happen here. We're just going to assume treatment of one eye, cost of treatment of one eye, and give the benefit from the study of one eye. And I don't think we can know more than that right now.
Thank you both. Tara, Dr. Lavelle.
Hi, thanks, Jason. I have a few questions, but I'm just going to see how long it takes to get through them. One question I had is the population that you modeled in the study, and I know that you relied a lot on the RESTORE trial to drive your inputs for the study. And so one thing like we always take away from these models is that all of this work is meant to inform population decision-making. But by relying on basically the inputs you derive from 27 patients. And I guess my question is, do you know, it seems very hard to determine or very unlikely that those 27 patients represent population demographics of the entire cohort of people that may be eligible for this drug in this country. And so what do we take away from this? That it's not cost effective in the RESTORE population, or then how do we then make decisions about the population, given how you're able to construct this model?
Yeah, thanks, Tara. And you're right. Like the small population is a key limitation. I will say, like our methods to deriving the inputs in terms of treatment effect was really a priori established that it was important to smooth the data and get rid of the noise of the impact of individual patients. And so that's what we believe we were successful in doing in this case. I would invite the clinical team if Neil has any comments on the generalizability. I think for the key inputs that mattered for this case was I think, well, patient age and starting level of visual acuity. And so I don't think we know enough about even the broader population in terms of that, because we received confidential data from the manufacturer who classified patients according to a level of visual functioning based on prior medical history, but we don't have a lot of insight into what that documented medical history looks like. And so I think that would extend more broadly even to a conversation we have already had about just not really knowing this population and how long they've been with RP or have been at the level of visual functioning that they are when they would receive the treatment.
Thank you.
Hey, Jason, do you mind? This is Sarah. I just want to quickly Tara, to your point, though, remember that what we're trying to do is help understand what a fair price is for society to pay for something based on the data that the manufacturer, who is then going to charge society, produced. And so we'll always have trouble with generalizability from a clinical trial to a bigger population, but that is the core of why we do this analysis to inform population level decision-making. Is it's the manufacturer using that data, and we're using that data to make a determination about a fair price for society.
Yeah, that just leads into problems with the manufacturer having the ability to have a trial with extreme selection bias, right? So there's just limitations to how these things end up being conducted. But yeah, certainly take the point that these were the best data available and you did absolutely the best you could with them.
I thank you all, and thank you, Sarah, for chiming in. Steve.
Great, thanks. Terrific job with the modeling as always, Dr. Richardson, and for your clear presentation. We all thank you. ICER's value-based price sort of solves for, what would be an acceptable or a fair price. In this example, we could also solve for, at that price, what degree of improvement would be necessary to achieve a quality benchmark, cost per quality benchmark. So, for example, just solve for how much utility gain would have to be achieved for the product to be considered cost effective. And you can do that in this case because there's no survival difference. So you can really focus on, that difference in utility. And you can almost kind of do it back of the napkin. And if you do, you need about 3 times as much utility gain to get to a cost-effective threshold that would be considered acceptable value. And to achieve that, then we're looking at utility gains about 0.2 points over 10 years or 0.25 points over 8 years, something like that. And the point is, it seems like with this approach, it's not doing the product any favors in terms of the degree of benefit that needs to be achieved for it to be considered to be cost effective. And I'd be interested in your thoughts about, that notion. And also, I think because of that, the outcomes also included cost per improvement on the other measures, which I thought were really interesting and different for an ICER economic analysis, but there wasn't a lot of talk about how to interpret what those outcomes represent or if they were acceptable or not. So sorry for such a long-winded question. Hopefully you can make some sense of it, and I'd be interested in your thoughts about all of that.
Okay, thanks, Steve. Okay, so a couple of thoughts. Yes, so no benefit in survival. So you're right, it all comes from health-related quality of life gains. I think one very big challenge as well that we haven't talked as much about is the lack of understanding about how quality of life changes across levels of visual functioning at such an advanced level of vision loss for this population. So we actually heard in some cases that once patients reach a certain level, like seeing hand motion, then no further progression would have an impact on quality of life. Now, I recognize that this is quite variable across patients we heard from, across the literature. And so we did do scenario analyses around that using different inputs. So I think it's just a challenge in general for this population to be able to show benefit across health-related quality of life. I think your second question, now I've lost it. Can you remind me?
Yeah, the other outcome measures, cost per improvement in the secondary outcomes.
Thank you. Yes, so another, I think a very telling, a nice complement to the health-related quality of life measures, which is important to note and very important to patients. So, even if we can't see as much benefit on health-related quality of life, we are seeing that patients spend, I think I don't have it right in front of me, but over 3 years in a better than counting fingers health state improvement compared to usual care. And so I think that should be considered for sure. The challenge is how do you incorporate that in, determining value in terms of cost or appropriate pricing. And for that, I mean, I think that I don't have any great solutions right now, other than let's talk about it, let's get it out there, let's make sure it's known to payers, to manufacturers, to the public that is also what matters to patients.
Thank you so much. I think we have time for, I appreciate everyone getting everything on time. We've kept on time. One more question from Joe Porter.
Yeah, thanks. And mine's super quick. I just want to make sure I'm understanding something in the slide. So like slide 58, it appears as though there was nothing anticipated about this treatment that would change the costs of care from normal treatment or usual care. So that there's nothing about this that changes how the costs would be anticipated from folks who were not getting this care. Is that correct?
That's right. That's in particular for direct medical costs.
Got it.
So for things like inpatient, outpatient, doctor's visits. That is slightly different for the societal perspective, which includes costs around changes to patient and caregiver productivity.
Got it. But nothing about this allows folks to spend less in their care for other.
Correct. That's what we found. Yep.
Thank you.
Thank you all so much, and congrats again to Dr. Richardson and the economic modeling team on a great report. We are right at 11:40, and we'll move now. We have a great